Welcome back to our discussion on reproduction and development. This is the second of a series of 3, so this is the B, number 2. And we are going to, as quickly as possible, have just a very cursory review, overview of the male and then the female reproductive structures, and then discuss the physiology of the system by gender. And the tradition is male first and then female. Leave the best for last. Is that what? Anyway, grew up with 6 sisters, so I guess I'm sensitized to those things. The external genitalia in the males, the penis and the scrotum, as we had said. The common passageway is the urethra. The accessory glands and ducts are the prostate gland, the seminal vesicle, and the bulbourethral gland. We had seen the features of the development of these two. The bulbourethral glands are added to the list and will undergo a similar type of a developmental process, but they are present in both male and females. They just have openings into either the space between the labia minora and the female, called the vestibule of the vagina, or directly into the base of the penis, the proximal portion of the penile urethra. This is the membranous urethra, where this urethral diaphragm is, urogenital diaphragm as it's called, the skeletal muscle. The external urethral sphincter is there, the voluntary one, and the involuntary smooth muscle urethral sphincter is here, as it would be found in the female, at the base of the urinary bladder, opening into the urethra. We see the prostate surrounding the prostatic urethra of the male, and the ejaculatory ducts opening into there, and those lead back to the ductus deferens that continues all the way to the gonad. So again, we considered much of this in the development, and beyond what I just stated, won't be discussing many features of this. There are 2 portions of the erectile tissue, a rigid type with a lot of connective tissue, and a spongy type, the corpus cavernosa for the former and corpus spongiosum for the latter, both of which are also present in the clitoris of the female. In the male, the corpus spongiosum completely surrounds the penile urethra, and therefore it's critical that it is spongy, such that the urethra can maintain patency. The corpora cavernosa are considered in the context of having cavernous venous sinuses with a central artery in each of them, and is the regulation of the blood flow in this type of erectile tissue. Again, it will be the same for the female. The corpora cavernosa and the corpora spongiosa are represented in the female for erectile tissue of the clitoris, and it is a physiological regulation of blood flow for the engorgement of this erectile tissue that produces the vasocongestion, as it's called, that generates the erection of either the penis or the clitoris. It's basically the same type of vasocongestion that you might experience if you get allergic rhinitis and you have congestion of your nasal passageways. We also notice that there's a dorsal penile artery paired and dorsal deep vein, dorsal superficial vein, and then, of course, the nerves. So the neurovascular supply, the lymphatics are not shown here, neurovascular supply is extremely important. It's just kind of funny. This looks like 2 big alien eyes and get away from that. The male reproductive structures, then, of course, the testes for sperm production are the context here. The seminiferous tubules contained inside the testes will be important. Those open through a connection into the epididymis, where the sperm will undergo maturation. So we're seeing functional anatomical subcompartmentalization of the male reproductive duct system, or tubular system. Sperm production in the seminiferous tubules within the testes. The first portion outside the testes, the epididymis, is where the final maturation will occur. And so with an ejaculation, this is the source of the mature sperm that have the capability of being motile to move through the female reproductive tract and reach the ovulated oocyte. The vas deferens, also known as the ductus deferens, is a passageway into the abdomen and delivery to the urethra. So from the testis, the seminiferous tubules within there, the developing sperm will reach the epididymis, a coiled tubular structure. So over a short overall length, there's a lot of space to house the nearly 200 million sperm or more. I'm not sure exactly how many ejaculations can be stored in there, but each ejaculation contains a couple hundred million, and the rate of production in the adult male, although it can change with age, is approximately 200 million a day. The authors go through that. Anyway, just thought I'd repeat it. So we keep it in mind that there's a certain number stored in the epididymis, and from that reserve, with an ejaculation event, they of course are projected outside of the body. And along the way, either the epithelial cells lining the pathways or accessory to it, like the seminal vesicle or the prostate gland, will contribute liquid contents to the cellular contents, the sperm that come from the epididymis with ejaculation, such that we have the collective solution, the semen, being ejected from the external urethral orifice with an ejaculatory event. The bulbourethral gland contributes a small amount of this as well, but this is typically the pre-ejaculate coming from the bulbourethral gland, but that does not mean that there is not already some sperm moving through this duct system, and we all know that the pre-ejaculate can contain sufficient sperm to cause conception and pregnancy. So we are going to pass through various levels of the gross anatomy to the microanatomy to look at just a portion of a seminiferous tubule. There are many of them, and they are highly coiled, and of course they join with this other tubular system that ultimately connects to the epididymis, and there's the head, the body, and the tail of the epididymis. If you sampled sperm from the head of the epididymis, they would not be very motile at all. But if you sampled from the tail, closest to the vas deferens, they will be fully motile and capable of all tasks required of them to make their way through the female reproductive tract and undergo conception to generate fertilization process. So now we're going to look at a small portion of a single seminiferous tubule here, just one single cross section, and then we're going to take a look at the highlighted portion here. What we see is that there's a connective tissue lining the outside of the seminiferous tubule, and that actually separates it from the vasculature. So there is a blood-testis barrier that is described in the textbook. It's more of an immunological barrier than a barrier to the movement of certain molecules. As we will see, some hormones are capable of passing across this. Of course, the steroid hormones freely diffuse, The water-soluble ones, like the gonadotropins, the FSH will bind to the certain cell population. We'll get away from those; they don't have it. The Sertoli cell here, the sustentacular cell, it sustains the sperm production process. So we think of this connective tissue capsule separating those cells that are within the seminiferous tubule from those that are outside of and between or among the seminiferous tubules. And of course these are the interstitial cells, but if you use the eponym, the name for the structure or the cell based upon the individual that discovered it, the Leydig cell. So the interstitial cells of Leydig are outside of the seminiferous tubules and between or among them, if you want to think of them collectively. Remember, between is just with two, and among is when there's more than two structures that you're talking about. So the neurovascular bundle is in the connective tissue in the area outside the seminiferous tubules where we find the interstitial cells of Leydig, so very readily are accessible to the anterior pituitary hormones, the gonadotropins. Okay, the spermatogenesis in the testis is what we want to consider on this slide, begin to consider. So the seminiferous tubules, as we saw, is where we'll find the Sertoli cells, and we'll go to that most zoomed-in picture, the highest mag illustration, in just a moment in the next slide. To kind of recap this, the Sertoli cells surround and support the developing sperm, as we saw. And we'll see again in the high mag, spermatogonia divide and produce all the sperm cells. And it proceeds from the periphery toward the center, the lumen of the tube. Right. So the spermatogonia are going to be here, undergo mitosis to replace themselves. If they didn't, then you'd run out of spermatogonia. And one of the daughter cells will go on to undergo the meiotic division to produce the gametes. So these spermatogonia divide and produce all the sperm cells. So with a division, a spermatogonium is going to regenerate a spermatogonium, a reserve cell, and then a primary spermatocyte that will go on to produce the secondary spermatocytes. Then finally spermatids that are immature, and then finally the spermatozoa that will undergo the maturation. And final point of maturation is the tail of the epididymis. The interstitial tissues support spermatogenesis. There is a term spermiogenesis that is part of the full spermatogenesis process, and that spermiogenesis refers to the final maturation of the spermatids to the fully motile and capable spermatozoa. So the interstitial tissues outside of the seminiferous tubules support the spermatogenesis. The interstitial cells of Leydig will be providing the testosterone. The capillaries deliver nutrients and remove wastes. This slide we see is the detail of the seminiferous tubule, as we had said. And we can orient ourselves to the interstitial location that is outside of the fibrous connective tissue capsule of the seminiferous tubule, and the interstitial cells of Leydig are there, as well as the complete neurovascular bundles, but we're just looking at the capillary bed here because we're thinking of the exchange of nutrients and hormones and waste and all the things that we've considered through the semester of study, but most importantly the hormones, right? So what we are proceeding to envision is the LH and FSH diffusing out of the capillary plasma and bouncing around in here and passing through this connective tissue capsule. And if there is a cell that expresses the receptor, like the Sertoli cells or the sustentacular cells, both the same name for the same cell, they will have the FSH receptors, and then these cells will express the LH receptors. So LH will target these cells. Even though it's diffusing everywhere, it can only affect the target cell. By definition, the target cell is the one that expresses the gene for the receptor and can bind the ligand, the hormone, and respond to it, and links the receptor to the responsive mechanisms. So remember your endocrinology. So this is the reason for showing this, and then picturing the development of the spermatozoa. See, and they're labeling these as spermatozoa, and they're really still this. They're the first ones. The spermatids are these on the innermost, and then these are the immature spermatozoa. Let's call it that way. They still have to undergo more spermiogenesis. So the metabolic changes within these immature spermatozoa are really what are going to change, because the anatomy is going to be the same whether they are immature or mature. So if you sampled right here and at the tail of the epididymis, the anatomy is going to be essentially the same. There might be some modifications, but it's the metabolism inside such that by the tail of the epididymis, this flagellum is capable of full operation. And so we see there's a head, and then I usually use the term the neck, and the midpiece, and then the tail. The term for the neck isn't really that critical. We can see in the head, of course, is the haploid nucleus, and the Golgi apparatus basically becomes this acrosome that is filled with enzymes essential for the final maturation stage, capacitation. There's an acrosomal reaction and everything, so that it can undergo conception and contribute to fertilization. The centrioles link the head with the midpiece, and this is where a lot of the ATP is needed, so you have the mitochondria just all spiraling around this structure that needs it tremendously. So quite obviously you have to have all the support structures for glucose uptake, glucose metabolism, anaerobic, and of course, because of the mitochondria here, the aerobic. So the motility of the sperm is very much dependent upon aerobic respiration, and we've talked about that in prior chapters. So let's consider the regulation of spermatogenesis. So the hormones control to the production of sperm. GnRH for the gonadotropins. The LH, as we said, targets the Leydig cells, the interstitial cells, to support the testosterone synthesis and secretion. FSH on the Sertoli cells to generate cell products. So these are also secretory cells. One of the secretions, they are paracrine cells. The signals will be contributing to or sustaining, because they're sustentacular cells, the sperm development. One of the secreted substances is an antigen binding protein that helps keep the androgen concentrations high in the lumen of the in the extracellular fluid compartment of the seminiferous tubule. Remember, the only way you can alter the local concentration of a steroid hormone is to have something there that will bind it dynamically, just to kind of hold it into place. So imagine someone standing in one of those glass chambers with money blowing around, and they're reaching out, and so as they're grabbing the money and trying to keep it close to their body, they are creating a localized increase in the concentration of the money, but otherwise it just escapes freely. I know that's contained, but anyway, it helps you to picture that this is the only way that you can produce a localized differential concentration in the lipid-soluble molecules, is to transiently occupy them at that point in space and time. As well as the same thing applies for the plasma proteins that help to keep a high concentration of steroids in the plasma, so you get diffusion out into the tissues. You can hold it there long enough to distribute it through the vasculature and finally let it diffuse into the tissues. And the Sertoli cells are also the source of inhibin that is going to produce the negative feedback on the FSH release specifically. So the Sertoli cells are expressing the FSH receptor, and part of the response to binding FSH to the FSH receptor is the synthesis and secretion of inhibin, so it contributes to its own negative feedback, if you want to think of it that way. And that's the way it should be. So the GnRH and the LH and FSH secretion are inhibited by the negative feedback of the inhibin and the testosterone. Well, let's look at it in this schema then. The hypothalamus and the anterior pituitary, glands 1 and 2, hormones 1 and 2, the GnRH and the FSH and LH, the gonadotropins. Looking at each separately, the FSH at the testis will be binding to receptors on the Sertoli, or the sustentacular cells. These are metabotropic receptors, so secondary messenger systems will be affected, bringing about change in cellular products. The cellular products will have local paracrine effects and sustain, as a sustentacular cell should do, not only a physical support but a metabolic support for sperm production, spermatogenesis. So again, remember that this label for spermatogonium is offset. It should be right over here. This is the Sertoli cell. The spermatogonium would be there and undergoes division, so it is more differentiated toward the center of the seminiferous tubule versus the periphery. So the basal cells are always where we find the reserve mitotic cells in these types of tissue structures and organizations. The inhibin is produced by the Sertoli cell and has the direct negative feedback. Another paracrine structure is the endocrine binding protein that will occupy testosterone and keep it at higher levels, and the testosterone has to be at higher concentrations here in order to have effects on these developing sperm cells. Now the Leydig cells, of course, we're going to get that testosterone there through the LH effect on the Leydig cells, so they express the receptors for LH, and through similar second messenger systems, cyclic AMP is one of the major ones, there will be the impacts on the biosynthetic enzymes to increase the synthesis throughout the pathway, plenty of cholesterol around, to feed into the system, and then ultimately testosterone will be one of the highest levels generated in the whole pathway. It can have effects on the Sertoli cell, freely diffuse, and of course be present to bind to the antigen binding protein and be available to have effects to sustain this whole process, as well as of course being washed away by the local capillary bed and carried and distributed to the entire body. And one part of the body, of course, is the hypothalamus and the pituitary, where they will contribute, the testosterone will contribute to the negative feedback. The accessory glands will contribute to the semen, as we had said. That's about 99% of the semen volume. So the seminal vesicles contribute fructose, vitamin C, prostaglandins. The prostate gland, interesting, the prostate gland does not contribute the prostaglandins. The buffers citric acid and the enzymes, and then the bulbourethral gland contributes mucus and buffers. We can then consider the composition of semen in a little more detail in this table. So the sperm function as the gametes, come from the seminiferous tubules. The mucus, the lubricant, from the bulbourethral glands. The water provides the liquid medium, comes from everything collectively. The buffers neutralize the acidic environment of the vagina, coming from the prostate and the bulbourethral glands. The nutrients will nourish the sperm, fructose, citric acid, vitamin C, carnitine. The seminal vesicles, the prostate, seminal vesicles, and epididymis will be contributing these respectively. The enzymes will form a clot of the semen in the vagina, then allow a liquefaction once deposited close to the cervix, and mixing with the cervical mucus, allowing the sperm to find their way into the uterus from the vagina. So the seminal vesicles in the prostate will contribute these enzymes. Zinc, we're not really sure what it might be doing. Possible association with fertility, and the source is unknown. Prostaglandins will affect the smooth muscle contraction and may aid the sperm transport, and they're coming from the seminal vesicles. The male secondary sexual characteristics are considered on this slide. So the secondary sexual characteristics are traits other than genitalia that distinguish males from females. So the general body shape, including the pattern of the muscle development, body hair and beard, deep voice, male sexual behavior or libido, the sexual drive. The androgens are called the anabolic steroids, and we are familiar with the illegal use of these. The female reproductive system, of course, is a cyclic process. The hormones are from the hypothalamus and anterior pituitary, and they stimulate the ovaries. So essentially the same three-gland, three-hormone system. The gonad and the hormones, three, will be different. The ovaries are releasing the estrogens and the progestins in varying patterns during each cycle. Ovulation releases a mature oocyte, one of the ultimate objectives of the cyclical release of the hormones. And then estrogen and progesterone prepare the uterus for the possible pregnancy, a second objective of this cycling hormonal system that I will call collectively the menstrual cycle. And we'll delineate that in a moment. The female reproductive structures, the external genitalia, also known as the vulva or the pudendum, the labia majora, labia minora, the clitoris, vagina, the hymen, the urethra opening inside the labia minora. This is improper English. We should convert it to the anatomical terminology, medial to the labia minora, or between. The space between the labia minora is the vestibule of the vagina. The external urethral orifice opens into the vestibule of the vagina, medial to each labia minora. An anatomist correcting that statement there. So this is the classic view from the perspective of a healthcare professional examining the external genitalia of a female. And of course, we recognize the anterior and posterior features. Anteriorly placed, the labia majora will converge at the mons pubis, and at the midline, projecting posteriorly from there, encounter first the clitoris with the folds of the labia majora, labia minora meeting anteriorly as well, but splitting to form the prepus and surround the glans of the clitoris that has both spongy and dense erectile tissue, the corpus cavernosum and the corpus spongiosum, in reverse order there. And continuing to move posteriorly, the external urethral orifice, or the urethral opening, this space between the, or medial to each labia minora, is the vestibule of the vagina, and the vaginal introitus, as it is called, or the orifice of the vagina, opens into this area as well. They're not showing it, but the bulbourethral glands open into the posterior portion of the vestibule of the vagina, secreting mucus as similar to the secretions in the male. So the female internal structures, the vagina, the uterus, so the vaginal introitus or orifice is the opening, and beyond that internally is the vagina, so the entire vagina from that opening inward is an internal organ. The uterus, the cervix, the endometrium and the myometrium, the fallopian tube, the fimbria and the ovaries, so these will be reviewed again in a very cursory fashion. So in a midline view of the human female, we see the clitoris. This is there's a glans and a shaft of the clitoris as well. They're showing here mostly the corpora cavernosa, but there is some corpora spongiosa contributing to it. And we are seeing the membranous urethra, where the external urethral sphincter is, and the internal smooth muscle involuntary urethral sphincter here, and the urethra is much shorter in the female, of course. Lower urinary tract infections are a greater risk. And the urethral orifice, or the external urethral orifice, opening into the vestibule of the vagina. So posterior to this is the opening into the vagina, and we can see the lumen of the vagina, and it of course folds over or beyond, the cervical opening, the external os of the cervix, the cervical canal, the internal os, the uterine cavity that will be continuous into the fallopian tubes, of course. So here you can see that there is a location which is a typical site of the deposition of the sperm, so that initial gel formation of the sperm and then liquefaction will be important to localize it near the cervical mucus secreted by the glands in the cervix of the uterus. And then this is important for orienting the sperm into the cervical, the external os, the cervical canal, and on through. You see also the association with the rectum and the anus, and here's the external anal sphincter and the internal anal sphincter located here. Now then, to divert attention briefly to the ovaries, considering the cells and the tissues there. So the follicle is the structure where the oocytes develop. So there are granulosa cells surrounding the gamete cell, the oocyte. There are thecal cells that surround all the follicles. So the theca is like a wrapping around the outside of the granulosa cells. And as the follicles develop, there would be a very critical association between the granulosa and the thecal cells for the synthesis of the different steroid hormones. And then ultimately the corpus luteum will be the remnants after the oocyte is ovulated. The remnants of the theca and the granulosa cells differentiate into the corpus luteum, a process called luteogenesis. So we can see this progressively occurring in this standard illustration of an ovary. The primary follicles, remember, are all that remain at birth. There are no oogonia, and each of these is surrounded by granulosa cells and a thin thecal connective tissue type of a capsule around them. It's not really that definitive like the thick connective tissue capsule that surrounds the gonads, but in essence it's kind of the same, in that they will be recruited and undergo development through the menstrual cycle. And the one follicle in humans that somehow is selected to complete the maturation process will have the generation of the granulosa cells and a thickening of the thecal cells, and these two will contribute to the formation of the gonadal steroids. And then of course, mature follicle, the liquid fills in this area here, and then the process of ovulation occurs with the rupturing of the follicle, and the ovulated oocyte is externalized. You can see, if you follow all the way from the opening into the vagina, this is the external environment, all the way to the inside of the peritoneal cavity of the female. So this is exteriorized. It is now in the external environment, and is, of course, picked up by the fimbria of the fallopian tube, and then carried up into the infundibular part, and then there's a little ampulla, a little dilation in the area right here, the most common location for fertilization, as we'll see in the next presentation. So this is an exteriorization of the mature oocyte. There's only one from one ovary in the humans. There's 2 ovaries, of course. So only one egg typically made available with each menstrual cycle. And then that single ruptured follicle will undergo the luteogenesis process to produce the corpus luteum, and then undergo luteolysis, or regression of the corpus luteum, if fertilization does not occur, or ultimately throughout pregnancy the placenta takes over the role of the corpus luteum. And these accumulate through life, and finally form a scar tissue called the corpus albicans. So the human female ovary accumulates corpora albican scars throughout life. Just a brief orientation to the uterus, which is going to be one of the target organs of the gonadal steroids. There's the outer mesothelial layer, called the mesometrium, then the myometrium, thick, smooth muscle wall, through which penetrate the neurovascular bundles. The arteries are the only ones showing here. The innermost layer is the endometrium on the inside. And we see that the arterioles end up forming these spiral arterioles through the thickness of the endometrium, and we know the endometrial thickness is going to change throughout the menstrual cycle, and of course throughout pregnancy. It is the nutritive source, or the site of exchange of nutrients and wastes for a pregnancy. The phases of the ovarian cycle are considered in this slide. I like to present the entire menstrual cycle as having 2 cycles within it, the ovarian and the uterine, as we'll talk about. And the menstrual cycle has phases as well. The ovarian and the uterine cycles have phases. The menstrual cycle I consider to have and all of them, of course, have that central phase that they're linked to, ovulation. So I consider the menstrual phase to have a pre-ovulatory phase, which includes the follicular phase of the ovarian cycle, and a post-ovulatory phase, which includes the luteal phase of the ovarian cycle. The authors give a slight variation of that. They say that the luteal phase is also considered the post-ovulatory phase of the ovarian cycle. I prefer to consider the menstrual cycle as being all-inclusive, having events before ovulation, the ovulation events, and then post-ovulation. So pre-ovulatory and a post-ovulatory phase overall. And then we look at the 2 separate cycles in this slide, the ovarian having a follicular phase corresponding to the and note I'm using the term corresponding to the pre-ovulatory phase of the menstrual cycle. So a subcycle of the overall cycle having a phase, the follicular phase, specific to it, to the ovary, that corresponds to the pre-ovulatory phase of the overall menstrual cycle. The reason for emphasizing that is that we will see that there are corresponding events and phases of the uterus for the uterine cycle. So we'll build on this idea as we go. So I will talk about pre-ovulatory phase of the menstrual cycle, or post-ovulatory, and then follicular phase of the ovarian, or events and phases of the uterine. So I will try to be emphatic in each of those cases. So the follicular phase of the ovarian cycle includes proliferation of the granulosa cells. So I like to remind ourselves that there is a recruitment and development of primary follicles in the follicular phase. And within those follicles that are recruited, there will be proliferation of granulosa cells. Remember, only one of them is going, a pool of primary oocytes will be recruited with each ovarian, with each menstrual cycle, and only one of those in one ovary will be selected to undergo the final maturation and capable of becoming capable of ovulating. So many are lost for the one to be supported to full maturation. So many primary follicles must be recruited and undergo proliferation to produce sufficient amounts of the gonadal steroid hormones to sustain whole organism levels to coordinate everything that's going to go on, and this only requires as an endpoint the maturation of a single one, because as you know, most commonly humans have single conception events. They will only carry one offspring with each gestation period. And there's multiples, but it's not the same as dogs having 6, 8, 10, 12 in a litter. So in humans, this is different. There's just one that will mature. That's the objective. It's never 100% perfect, but that's the objective. With drug treatments, you can force multiple ovulations, and this is what will happen with fertility treatments to collect mature oocytes for in vitro fertilization. Okay, so at ovulation, that ripened follicle, the mature follicle, will rupture or burst and release the oocyte as we saw. The luteal phase of the ovarian cycle, corresponding to the post-ovulatory phase of the menstrual cycle, is where we follow the ruptured follicle, the granulosa cells and the thecal cells as they differentiate into the corpus luteum. They undergo luteogenesis. And we'll see differences in steroid contributions to the steroid profile as we proceed through this. So the one phase that is constant across all three cycles in my model is ovulation. There's ovulation in the menstrual cycle as a central point, in the ovarian cycle as a central point, and in the uterine cycle as a central point. So we'll use that as a central point. It is typically shown as the center day in an average 28-day menstrual cycle in the human female. But this is the average, so I think 25 to 35 was given as the range in the textbook. We notice that it does vary. And at this time where we're talking about it varying, I will draw your attention to the fact that the corpus luteum typically lasts or survives, unless there's a pregnancy, 14 days. So from the first day of a menstrual flow, minus 14 days, a person can predict rather reliably that they ovulated on that day that already passed 2 weeks ago. The pre-ovulatory phase of the menstrual cycle, which is corresponds to the follicular phase of the ovarian, is the one of variable length. So if you are a female that has a 30-day menstrual cycle, your post-ovulatory phase is still 14 days, and the 2 days are added to the pre-ovulatory. So if you started at day 0, the first day of the appearance of menses from the vagina, then it would be 16 days to ovulation, and still 14 days for the survival, an additional 14 days for the survival of the corpus luteum. This is very important fact to understand about the menstrual cycle. Okay, let's draw our attention back then to this central point, ovulation. What I don't like about these illustrations is that they only show one follicle developing. We know that many are recruited in both ovaries, but only one in one of the ovaries will undergo this process that we are seeing here. So in other words, this single follicle undergoing development to become the mature follicle, it will happen in one case with each menstrual cycle, but this one alone, with the granulosa and thecal cells surrounding it, is not responsible for these total steroid and other hormone products that can be found distributed throughout the organism across these number of days. So that's why many need to be recruited, and only one is selected for maturation, a process that is still not fully understood, how we select just one. So the pituitary hormones, the gonadotropins, are shown in the upper panel here for the ovarian cycle of the menstrual cycle. And we are looking at the cellular events in the middle panel, and then the hormonal events. These are plasma levels being reflected in this illustration of the ovarian hormone levels. If we were sampling them from the interstitial fluid of the ovaries, we would expect to see a similar profile, but the post-ovulatory profile would only be present in the one ovary where ovulation occurred, and the single corpus luteum would have developed. So remember, these post-ovulatory plasma, in particular progesterone levels, are produced by one corpus luteum and one ovary, whereas all the other plasma profile hormonal events are generated by the full collection of the recruited primary follicles that persist up to ovulation. After ovulation, they begin to undergo a process called atresia, where the cells undergo apoptosis and are destroyed. And so after ovulation, virtually all of the hormonal profiles are generated by the single corpus luteum in a single ovary. So they are big, and they are loaded with lipids, and therefore yellow, because these lipids and cholesterol are needed to sustain the metabolic activity, and in particular the cholesterol to produce the vast amounts of steroid hormones. Okay, so let's follow the gonadotropin levels in pre-ovulatory and then post-ovulatory phases and at ovulation, and see the cellular events, and then try to correlate that with these, and we'll go through separate slides that will look at this in detail later. So early in the follicular phase, we have the gonadotropins that are going to be responsible, in particular, the higher level here is FSH, that's going to recruit and contribute to the development and cause the development of the primary follicles, all the ones that were recruited. Somewhere along that process, one of them will be selected to undergo the final development to the mature or Graafian follicle that you may have heard of. As the time progresses through the pre-ovulatory phase of the menstrual cycle, estrogen levels are going to increase dramatically as more and more of the developing follicular cells, all the follicles that were recruited, are just pumping out estrogen, so the estrogen levels are going to increase on average each day. So the daily average level is kind of what's represented here. Morning, midday, and evening might be variable, circadian, or minute to minute, ultradian. But anyway, each day being represented here as an average plasma level. It's going to be ever increasing to a high point at about a couple days before ovulation. This corresponds to that switch of negative feedback to positive feedback of estrogen that we mentioned in an earlier slide, and therefore we see the gonadotropin levels have increased. This LH and FSH surge, but in particular this LH surge, is essential for ovulation. While we're looking at it on this slide, if you introduce a higher level of exogenous ovarian female gonadal steroids, like the birth control pill, early in the phase, the preovulatory phase, it will ultimately prevent the LH surge, and it prevents ovulation. And this is how it acts as a contraceptive. The gamete is not available for conception. The endometrial lining of the uterus is going to undergo another a cycle as well. I almost said independently, but that's not true. It's completely dependent upon the hormone changes that occur in the from the ovarian cycle. But it is a separate cycle that can be considered anatomically and functionally as distinct from the ovarian, with its own cellular events. Doesn't produce hormones, but does have the cellular events. So the first, at the beginning, corresponding to day 0 of the menstrual cycle, is the menstrual phase, where menses will flow from the uterus through the vagina to be exteriorized, and it of course happens if there is no pregnancy as a result of that cycle of producing the single gamete capable of fertilization and conception. Bleeding from the uterus as endometrium is shed. So the endometrial lining is shed, and some of the blood that is contained within it. The endometrial cells themselves have a similar color to blood cells. The proliferative phase will be subsequent to that, to regenerate what had been shed. So these endometrial cells undergo tremendous changes to prepare themselves. They become packed with glycogen and fatty stores available for use of the developing embryo and fetus, and to establish the anatomical relationship between maternal blood flow and the placenta, as we'll see in the next lecture set. And it is a transient reserve. It isn't permanent in humans, in placental animals that undergo that have menstrual cycles, and therefore it cannot be sustained. So it has to be shed and then regenerated in the hopes that the next cycle will result in a pregnancy. So the proliferative phase, and then finally the secretory phase in preparation, in anticipation to implantation of a zygote, as it would be called, or concepsis, the result of conception. So the proliferative phase will correspond to the premenstrual phase of the menstrual cycle. The proliferative phase of the uterine cycle will correspond to the premenstrual phase, the pre-ovulatory phase of the menstrual cycle. The post-ovulatory phase of the uterine cycle corresponds to the post-ovulatory phase, the secretory phase of the uterine cycle corresponds to the post-ovulatory phase of the menstrual cycle. And here the endometrial secretions from the glandular portion of the endometrium will promote the implantation process. Well, let's look at this collectively then. So again, ovulation. So the ovulatory phase I introduce across all of these cycles. We're looking at the ovarian hormone levels as representation of the ovarian cycle. And we said these are responsible for the cellular events, tissue changes in the uterine cycle. So if we take it all the way back to the menstrual cycle, there is the pre-ovulatory and the post-ovulatory phases. The pre-ovulatory phases of the uterine cycle, the phases of the uterine cycle that are in the pre-ovulatory phase of the menstrual cycle are the menstrual phase, where menses occurs, and the proliferative phase. The uterine phase, the secretory phase that corresponds to or occurs during the post-ovulatory phase of the menstrual cycle, another way to say it, is this the secretory phase. And this is in anticipation of a post-ovulatory implantation of a concepsis. We had talked about with the thermoregulation, the body temperature changing, the basal body temperature changing in females in the pre-ovulatory versus the post-ovulatory phases of the menstrual cycle. So what hormone is highest during the pre-ovulatory phase? Estrogen. So the estrogen is responsible for the proliferative phase. You can see the proliferation almost follows the same shape of the curve here. They are all low in the early part of the pre-ovulatory phase, or the follicular phase of the ovarian cycle, and therefore they do not support the endometrium, and it is lost as menses. And then the estrogen levels increase, and this triggers the proliferation. After ovulation, those estrogen levels drop, and the corpus luteum begins to produce predominantly progesterone, and it promotes gestation, so it triggers the transition from the proliferative. In other words, proliferation ends when the estrogen levels start dropping, and then the secretion of the endometrium begins with the ever-increasing progesterone levels. Implantation would be occurring at about 6 days after fertilization, and so you can see that at the peak progesterone plasma level is about when you would expect implantation to occur, and at about that same time the concepsis will begin synthesizing and secreting what's called the human chorionic gonadotropin. And the human chorionic gonadotropin will sustain the corpus luteum, and then therefore with pregnancy progesterone levels would remain high, and they'd sustain the endometrium. So we're going to go through each of these phases collectively and try to put this together. So and then later we'll look at the neuroendocrine function. So in the early follicular phase, you can see the gonadotropic hormone levels, FSH and LH, with the recruitment and development of the follicles, and therefore the follicles are only being recruited and starting to develop, so the hormonal levels are going to be low, estrogen, inhibitin, progesterone, so they cannot sustain the endometrium, so the endometrium is going to be lost during the menstrual phase of the the uterine cycle. So here you see the phases of the ovarian cycle and the phases of the uterine cycle. So they use the term menses. I refer to it as the menstrual phase of the uterine cycle. The collective set here is the menstrual cycle. And the body temperature is being traced as well. As the recruited follicles population in both ovaries develop, more and more estrogen can be synthesized and therefore freely diffuses into the plasma and local tissues, and the average daily concentration is going to be ever increasing up to the approaching midpoint of the menstrual cycle, the peri-ovulatory period. Peri, I said, around ovulation. Or late follicular phase, as we'll call it later. And this is the switch from negative to positive feedback, and we get the surges of the gonadotropins, and these are critical for ovulation. And at ovulation, all the other follicles that didn't ovulate will begin to die off, undergo atresia, become atretic, and the cells undergo apoptosis within them, and they no longer contribute to the hormonal levels. And you're seeing the inhibin profile following that of the FSH to keep it under control. And therefore, the one corpus luteum that develops is going to take over the hormonal production, and you can see it takes a little time for it to build up, and as it is moving through the first half of the luteal phase, corresponding to the secretory phase of the uterine cycle, it's generating ever increasing levels, and then from that point on it begins to taper, and of course that's because we've switched back to the negative feedback. So here is negative feedback, this is positive feedback, very episodic, with immediate switch back to negative feedback as the estrogen levels begin to drop. And then when the progesterone, the hormones 3, especially progesterone, and then the estrogen comes back up, not as high as this, right? So it stays in the negative feedback range. We now have, but it has this progesterone contributing to it. We have peak negative feedback here. If there isn't a pregnancy to bring the HCG in to sustain the corpus luteum, then this negative feedback is going to eliminate the gonadotropins support in the corpus luteum, will undergo luteolysis, which is the same thing, the apoptosis. Without the sustained gonadotropin support, apoptosis will occur in these cells, and it just becomes the scar tissue. The corpus albicans would be the end point here, with no hormones coming from it. So I call the corpus luteum a suicidal structure. It ends up producing substances that destroy its supportive supply of gonadotropins, and it accomplishes this within almost exactly 14 days. So as I said, day 28, day 0, minus 14 days is the day that ovulation occurred. Projecting forward from the first day of menses is very unreliable. The complex hormonal control of the two cycles, the ovarian and uterine cycles, are controlled by several hormones which display complex interactions. So there are extra ovarian hormones, the GnRH from the hypothalamus, the gonadotropins from the anterior pituitary, the ovarian hormones that we had followed and tracked. And we're going to look at them now with each phase in a little more detail, and I know it's a repeat, but it doesn't hurt to go through it one final time here. So the hormonal control of the menstrual cycle, the follicular phase is what we are considering. FSH stimulates the follicular development, so recruitment and development. maturation to the secondary and the tertiary follicles. Only one will become the mature follicle. The granulosa cells produce the estrogen. The negative feedback decreases FSH and LH secretion, and there's a positive feedback that allows the estrogen production. And actually there's an interaction between the granulosa cells and the thecal cells for the production of the steroid. I don't want to mislead you here. The cells of the theca contribute to this process. It's called a 2-gonadotropin 2-cell steroid hypothesis. But anyway, it's just fine to add thecal cells. Granulosa thecal cells contribute to the production of the estrogen, and the negative feedback will decrease the FSH and LH secretion, and then there's the switch to the positive feedback that allows the continued estrogen production. The LH stimulates the thecal cells to produce the androgens. Aromatase converts androgens into estrogen, and the antrum fills with fluid. So here we are looking at this follicular phase. It can be looked at in the early and the late follicular phase, or just the peri-ovulatory phase. So we'll look at the early to the mid follicular phase collectively here. So you can break it down into a day-by-day series of events almost, but we're going to look at the early to the mid. So the GnRH from the hypothalamus, the gonadotropins, and then the follicle, the granulosa, and the theca cells. So here you see the 2-gonadotropin 2-cell steroid synthesis hypothesis is represented. LH targets the thecal cells, FSH targets the granulosa cells. The granulosa cell is the one that ultimately produces the estrogens, and that's why you have this statement here: the granulosa cells produce the estrogen, but I said the granulosa theca cells contribute to estrogen production to represent this collective concept here. So think now of what cells were the target of FSH in the male and what were the target of the LH in the male. We now have two different cells as separate targets in the female, but they are contributing, their cohorts, in the production of the female gonadal steroids. The thecal cells produce the androgens with the LH drive. The FSH drives the granulosa cells to produce the estrogens, and remember, it's aromatase is a key factor here. The estrogens have a positive feedback on the granulosa cells, right in this area. But in general, at the lower levels here early, the negative feedback. So it's this, the early to the mid follicular phase. If you yourself, or if you know of someone that takes the oral contraceptive pill, basically you stop it one day to end a menstrual cycle. This triggers menses, and 7 days later, correct me if I'm wrong, don't follow me and get pregnant here, but anyway, roughly a week later you start taking them again, or you take a placebo, a sugar pill in between, right? Depending upon how it's administered. But basically, the external steroids regulate the menstrual phase of the menstrual cycle, how long the menses phase will be allowed, and then they're contributed. So basically, this transition from early to mid-follicular phase is when the exogenous steroids are added, and they take advantage of this very sensitive negative feedback at the level of the pituitary and the hypothalamus. And what they do is they stop the progression right here at this point. Estrogen levels don't get high enough. They don't get any higher than a little bit that's synthesized and the exogenous supply provided by the oral contraceptive pill. They never get high enough, as we'll see, in the late follicular phase to trigger the positive feedback. And they prevent the LH surge. So the hormonal control, so that early enhanced sensitivity to negative feedback is very important in that type of contraception, the oral contraceptive pill. The hormonal control of the menstrual cycle continuing, the late follicular phase and ovulation. So during the late follicular phase, the secretion of the inhibin and the progesterone begins. The estrogen changes from the negative to the positive feedback. The enhanced secretion of LH leads to the LH surge, critical for ovulation. If this is prevented by preventing the production of high levels of estrogen and the switch from negative to positive feedback to the mechanism we just talked about is one example. If you block the LH surge by that means or any other mechanism, ovulation does not occur. Meiosis resumes in the developing follicles. Then at ovulation, the mature follicle secretes collagenase. The inflammatory reaction produces prostaglandins. The follicle ruptures, releasing the mature oocyte. The theca and granulosa cells change into luteal cells, the luteogenesis that we talked about. Now, remember, I criticize this presentation because we have many follicles recruited and developing. All of them except one in humans will undergo atresia at this point just before ovulation or around at ovulation, the peri-ovulatory time period. And the one follicle that undergoes ovulation is the only one that undergoes luteogenesis. So we go from many to one, but that one becomes very big. The corpus luteum, relative to the size of the secondary or tertiary follicles that don't go to maturity, that undergo atresia, a single one of those versus the corpus luteum is a tremendous size differential. So the late follicular phase is shown here. At this point, inhibin is beginning to regulate how much FSH can be synthesized and released. It's having a more pronounced negative feedback effect because more of it's coming from the granulosa cells. And the high estrogen output is transitioning that feedback into a positive feedback. And there's small amounts of progesterone that are also now going to have a positive feedback effect. But otherwise, we still have everything as it was. Targets don't change. Androgens coming from the thecal cells, diffusing to the nearby granulosa cells, and either being converted to small amounts of progesterone or high levels of estrogen. So the hormonal control in the menstrual cycle, early to mid-luteal phase is what we want to consider next. But let me back up just one point here. What we need to think about are two mechanisms interacting to begin to produce high levels of estrogen and shift to have more significant amounts of progesterone being produced such that they can contribute to the plasma concentration of progesterone that's detectably higher. One of those is you have to have increased gene expression to have more of the enzymes around, and then those enzymes become activated by the secondary messenger systems, mostly the cyclic AMP system, from the FSH. These two together, the enhanced gene expression so that there's more enzymes, a greater biosynthetic capacity in both of these types of cells, enhanced gene expression, more of the enzymes available to be activated by the second messenger systems, and there's sufficient amounts of the gonadotropins to drive the receptors to activate more of the enzymatic proteins, molecules that are present. All you need to do for the steroids is have more enzyme activity, and they freely diffuse away as they are being produced. So you need to change certain populations of enzymes so that more progesterone is produced at any one point in time and available to diffuse away and contribute to a plasma level that's higher. and then tremendously amounts dedicated, especially aromatase, to converting anything that's available into the estrogen so it can diffuse away and contribute to this very high plasma estrogen level. Remember, what you're seeing reflected in the plasma for these steroid hormones in particular is reflected for every tissue because they go everywhere, and unless you've got proteins to occupy them and generate a localized high concentration, they will equilibrate. So in other words, the amount of estrogen in the skin of a female during the periovulatory time period is going to be higher than the amount of estrogen you'd find in the skin cells during the menstrual phase of the menstrual cycle, because it diffuses everywhere. The hormonal control can only have effect on cells that have the receptor expressed, of course, but it's still going to be there. The hormonal control of the menstrual cycle early to mid-luteal then. So we have bypassed ovulation, and now the corpus luteum develops, continues the luteogenic process, and secretes progesterone and estrogen. So a tremendous shift in the expression of genes for all of these enzymatic proteins and everything else to make and store the fats and trap the cholesterol to esterify it and things like that, and to soak up cholesterol from the plasma to pump out these, especially progesterone. Very high synthesis rate of both of these, but much higher for progesterone. Progesterone and estrogen exert the negative feedback in the hypothalamus, so as we said, by about day 20 or 21 of the average menstrual cycle, you have peak levels of progesterone and estrogen in the mid-luteal phase, and you're going to begin to shut down the hypothalamus and pituitary and terminate the menstrual cycle collectively. So the effects of the progesterone on the endometrium contribute to its final development and the secretory nature of it. There's secretory glands within it. The cervical mucus will thicken. So what wasn't mentioned is that the estrogen will thin out the mucus in the cervix close to, so that where you have the high estrogen levels, you'll have the thinnest mucus secretions with strands of thicker mucus within it, and these become the little channels for the sperm to follow to find their way to the external os of the cervix. So at all other times, the cervical mucus is going to be thicker. You only want it thin at the time when the sperm are anticipated to penetrate and pass to the deeper structures. So here we see, again, our illustration, the corpus luteum. High levels of progesterone especially, but also of estrogen and inhibin, and pushing toward the very sensitive negative feedback to shut down the hypothalamus and the pituitary, and ultimately it will do that suicide cycle that I said that takes about 14 days, so that at the late luteal phase, if there is no implantation, the corpus luteum undergoes apoptosis after 12 days. Lack of progesterone leads to the death of the endometrium. This is also how the progesterone receptor antagonists, RU486 is a name for it. I forget what it's being called for the prescribed drug. But it is now legal in the United States, so it can be taken at any time through the menstrual cycle, blocking the progesterone receptors, leading to the death of the endometrium, triggering menses, the sloughing of the dead endometrium. So at all times through the menstrual cycle, progesterone receptor stimulation is essential for the maintenance of the functional endometrial layer. The basal layer always stays and is never sloughed. It's the functional layer that's generated with proliferation and becomes secretory. that is lost. Those cells depend on progesterone presence at any time. They depend on estrogen to proliferate, but to stay there once they're made, they need the progesterone throughout. And therefore, in the late part of the luteal phase, when progesterone, the corpus luteum's gone, so progesterone's gone, and physiologically then the progesterone receptor is not occupied in these cells, they die, and the sloughing ensues. And of course, as the progesterone, as hormones 3 drop, FSH and LH secretion is going to begin to pick up again. So following implantation, actually even building up to implantation, the HCG levels will begin to be sufficient to contribute to corpus luteum maintenance, but at implantation, 6-7 days after fertilization, closely correlated to the day of ovulation, as you might know, there would be sufficient HCG levels to maintain the corpus luteum independent of the hypothalamus. And this is the way that also this negative feedback that we talked about up here that terminates a menstrual cycle is also critical for the pregnancy becoming independent of the maternal pituitary. So if for whatever reason a pregnant woman's pituitary gland, anterior pituitary gland, had to be removed, it could be removed without any threat other than the surgery itself to the pregnancy, because the pregnancy is independent of the maternal pituitary. Breastfeeding subsequent wouldn't be very successful because that is dependent upon the paternal pituitary. But anyway, the corpus luteum secretes the progesterone and the estrogen, as we said, and we can see that here. So they have dropped, and the negative feedback is removed. We took our foot off the brake pedal, and we begin the cycle all over again. Female secondary sexual characteristics considered on this slide. They are traits other than genitalia that distinguish females from males. Why do people always put the male first? Anyway, it's not that I am macho or not macho, it's just, anyway, a little disconcerting. The characteristics controlled by estrogen are breast development, distribution of body fat. The adrenal androgens control some female traits. Well, the author of the textbook is also female, so it's just kind of interesting. Maybe the author of this chapter is male. I'll have to go and take a look at that later. Anyway, gender discrimination within science, is that what it reflects here? The adrenal androgens control some female traits, the growth of the pubic and the axillary hair, and the sex drive, the libido. So, as we said, androgenic steroids are present in both genders, both sexes, and they do contribute